yeah, it's incredible.
I think it's a good place to actually ask sort of the big question that people might have is how do we know every aspect of this that you described is safe?
At the end of the day, the gold standard is to look at the tissue. Um, what sort of trauma did you cause the tissue, and does that correlate to whatever behavioral anomalies that you may have seen? And that's the language to which we can communicate about the safety of inserting something into the brain and what type of trauma that you can cause. So we actually have an entire department of pathology that looks at these tissue slices. There are many steps that are involved in doing this. Once you have studies that are launched to with particular endpoints in mind. At some point you have to euthanize the animal, and then you go through necropsy to kind of collect the brain tissue samples. You fix them in formalin, and you like roast them, you section them, and you look at individual slices just to see what kind of reaction or lack thereof exists. So that's the kind of the language to which FDA speaks, and as well for us to kind of evaluate the safety of the insertion mechanism as well as the threads at various different time points. Both acute, so anywhere between 0 to 3 months to beyond 3 months.
So those are kind of the details of an extremely high standard of safety that has to be reached.
Correct.
FDA supervises this, but there's in general just a very high standard. And every aspect of this, including the surgery. I think Matthew McDougall has mentioned that like the standard is, let's say, how to put it politely, higher than maybe some other operations that we take for granted. So the standard for all the surgical stuff here is extremely high.
Very high. I mean, it's a highly regulated environment with the governing agencies that scrutinize every medical device that gets marketed, and I think it's a good thing. It's good to have those high standards, and we try to hold extremely high standards to kind of understand what sort of damage, if any, these innovative emerging technologies and new technologies that we're building are. And so far, we have been extremely impressed by lack of immune response from these threads.
Speaking of which, you you talked to me with excitement about the histology and some of the images that you're able to share. Can you explain to me what we're looking at?
Yeah, so what you're looking at is a stained tissue image. So this is a sectioned tissue slice from an animal that was implanted for 7 months, so kind of a chronic time point. And you're seeing all these different colors, and each color indicates specific types of cell types. So purple and pink are astrocytes and microglia, respectively. They're types of glial cells. And yeah, the other thing that people may not be aware of is your brain is not just made up of soup of neurons and axons. There are other cells like glial cells that actually kind of is the glue and also react if there are any trauma or damage to the tissue.
But the brown are the neurons
The
here.
brown are the neurons and the blue is nuclei. So what you're seeing is in this kind of macro image, you're seeing these like circle highlighted in white, the insertion sites. And when you zoom into one of those, you see the threads. And then in this particular case, I think we're seeing about the 16 wires that are going into the page. And the incredible thing here is the fact that you have the neurons that are these brown structures or brown circular or elliptical thing that are actually touching and abutting the threads. So what this is saying is that there's basically zero trauma that's caused during this insertion. And with these neural interfaces, these microelectrodes that you insert, that is one of the most common mode of failure